Right, so I have about 15 minutes, I should mention, to walk you through the search strategies. And we already started to look at the search strategies through a Mars presentations, and you kind of got a feel to how you can use the resources to create searches using the exercises that we have just finished with you a few minutes ago. So I will use FungiDB interface, but the approaches or sort of logic to building searches is applicable to any of the ViewPathDB sites. And what I would like to do is to demonstrate how you can create a search strategy where you utilize resources in better studied organisms for which there are more data sets available, to gain some interesting insights into, let's say, new and emerging pathogens. For example, let's imagine for a second that we are Candida metaphylosis researchers, and we're interested in learning about genes that may be important for the disease, or some of the genes that are upregulated in response to exposure to blood, as to simulate how when Candida albicans or other organisms grow in human body. And so when I have looked at some of the data sets in FungiDB, I didn't find all of the data sets that fit that criteria. And so my job is to create a search strategy where I use all of the wealth of the information from other species to learn interesting and new information about the genes in my organisms. So why Candida metaphylosis is interesting, or why would you want to study? It's an organism that often causes infections. They're associated with healthcare conditions, such as if you stay in the hospital, you sometimes may pick up this from the hospital conditions. So let's take a look first at the data that is available in FungiDB. I will first look for any genes that are annotated with a disease phenotype in Candida. And I will do this by navigating to the search menu at the top. Now, the search menu essentially provides you with a list of all searches that we offer for all different kinds of data integrated in FungiDB or ViewPathDB in general. And the searches are categorized based on the data type records that they access. In this case, I'm interested in genes, so I would want to identify genes that have a phenotype, and the phenotype should be associated with anything that's related to the candidiasis, or a disease called by Candida species. So in my experiment, I will use records curated by Phibase, and Phibase stands for Pathogen Host Interactions Database, and they specifically aim to curate records relevant to virulence and disease phenotypes. And so if I click on the CP, which stands for the Curated Phenotype Search, I'll be redirected for all of the different data records that are housed within this data set. Now, when we integrate records from Phibase, we also pulled in their annotations for gene annotations for any disease phenotypes. And so in this case, I'll go ahead and I just close the filter because I've run the search and it just remembers my settings. So I'll start from scratch, but I'll go ahead and use the filter menu to look for anything that may be related to Candidemia. And so in this case, I'll just select as blanket to return any genes that may be relevant to this disease. And now I'll go ahead and click Get Insert. So notice that my search returned 390 genes, and in this case, I am selecting all Candida species. And if you're just curious about why the search is slightly different from the above, it's just because I selected 2 species here. So we can mimic this by essentially navigating and looking through the organisms that are represented in my search. Now, I noticed there was Candida albicans, but if I wanted to look up in greater detail, I know that there was 251 records for Candida albicans. I will go ahead and select that. Anything for Candida albicans, and I also look for Candida glabrata. Glabrata has been recently renamed, so you may know it by Necrocemyces glabratus, and as such, I will pull these records as well. So here's one way how you can filter your records based on the organisms that you would like to have in your search. I will apply this selection to my search, for example, or I can always change my criteria at some point later. But the bottom line is now that I have closely to slightly over 350 genes that are annotated with a Candida disease phenotype. And so the next thing that I want to do, I want to identify genes that are also maybe responding to infection when Candida species are grown in blood. We have several data sets matching this criteria, and because I have two organisms now, well, two species that are selected, Candida albicans and Candida glabrata, I'll try to pull the information for these data sets. So what I would do, I would click on Add Step, and so here you are trying to expand your search strategy and add additional steps to grow your in silico experiment. And in my case, and I will be using combined with other genes option, because I'll be looking for the genes that are upregulated in certain conditions. And in this case, we will be using the RNA-seq evidence search. As Amar has shown previously, there are several ways to combine different types of records within the search. Because our step 1 and step 2 are utilizing genes, we will be using the first option. So if I navigate to the RNA-seq evidence page, there is a list, quite extensive list of data sets for different organisms, including Candida. And if I want to quickly identify those that's applicable to my conditions, such as where Candida organisms are grown in blood, there are several come up for this search. So in this case, I will use the first data set, Candida albicans, to identify any genes that are differentially expressed. And in this case, I want to identify genes that are upregulated at the 240-minute mark, comparing to the 0 minutes or control. And I will choose upregulated and leave every other selection criteria at default. So when I run step, my search will return that I have 63 genes that are matching my criteria, meaning that they're upregulated in Candida and they also have a disease phenotype. But remember, I have two species in my first step. So we selected the phenotype Candidiasis, and I also have limited to 2 species. So in this case, what I would like to do is to add additional RNA-seq data to my experiment all in one step. And you can do this by growing your strategy as a branch rather than as a linear progression. And to do this, we have quite a useful option that is called Make a Nested Strategy. So this feature is available under a pop-up menu. If you click on the Edit step within your strategy, navigate to the top menu and select Make Nested Strategy. So what this step will do, it will open up a pop-up window where now you can create an additional strategy or sort of as a branch that will feed into the step 2. And in a particular case, I want to add another data set that is also RNA-seq data. But in this case, I also want to bring in a data set where Candida glabratus or Mycosiomyces glabratus was grown in blood. So I will also select differential expression because this is the same data set that use the same settings as the previous one. So everything else is identical, and I leave all the parameters that is the same. And I'll click on Next Step. So now notice that for some reason, I have all of these results, but I returned 0 genes. And that's because when quickly I was setting up my steps, I forgot to actually specify the Boolean operator that is needed to return a cumulative result from both of these searches. And to modify the Boolean operator, I can click on the button that says Edit, that is highlighted in yellow, and select the appropriate operator. In this case, I would like for my strategy to return results as a union, where I'm looking at the combination of the genes that are upregulated in Candida albicans and also in Candida glabrata, in response to growing in blood. And then subsequently take these results and insert them into the step 2 and essentially cross-reference these results with any of the disease phenotypes that I have identified based on the 5-base manual curations. And so at this step, you can click on the X button here to collapse the search strategy window. That doesn't delete the step, it just simply collapses it so it's easier to view. So at any point, you can click on the edit button within the step again and say show nested if you would like to remind yourself the steps that you have created here. You can also rename any of these steps at any time. So for example, here I could say edit, rename strategy, and my name will be RNA-seq for Candida albicans and Candida glabrata. And I will just click on the accept button and my step has been renamed. So notice that within the nested strategy search, we also have a slightly different appearance of the window. So the regular step before that utilized a single data set just sort of doesn't have any additional outline. So these outlines simply indicate that this step contains multiple steps, and as such, it is a nested strategy that consists of 2 or more steps. Now, coming back to my original question, I am a Candida metaphylosis researcher, and I wanted to basically to transform these records now somehow in my organisms, because these data sets, such as Candida metaphylosis growing in blood, is not currently available in FungiDB. And I can do this using an additional step. So I will go ahead and click on the add step. And in this case, I'm interested in transforming my records, right, into different species. So now I can select the step number 2, which is here listed on the left. It says transform into related records. This step is associated sort of with a different outline of the step of how it is appearing in your search strategy to let you know that you are transferring your records into different types of data. And in this case, the different type of data is ortholog information. So what I want to do is to transform 73 genes from the step 2 into orthologs in Candida metaphylosis. So what I could do is quickly identify Candida metaphylosis based by the filter, just cite a few keywords. And I have an option to look for either synthetic orthologs or those only for synthetic orthologs, where you would have a matching position across chromosomes. But because the metaphylosis is a little bit more divergent from Candida albicans and glabratus, I would say, identify any orthologs that is matching to the 73 genes in step 2. And I'll go ahead and run step. And so in this case, our analysis identifies that I have 129 genes that match this criteria. So it's quite a bit more compared to the 73 genes that I have returned from the previous step. And that simply could mean that some of these could arise from duplication. There is a divergence. And so in itself, it's an interesting observation to where now you have a list of proteins. Some of them will be hypothetical, which means there very little is known about the function of these genes. And some of them are annotated based on orthology from other species. So in this case, as a Candida metaphylosis researcher, you may want to download the list of these genes and explore this a little bit further to see if you could pull up some interesting domains, whether they're predicted with AlphaFold or annotated by other community members on gene record pages, etc. And I'm almost at the hour, so I just wanted to briefly mention that search strategies are also associated with additional enrichment analysis, which you will look in greater detail in subsequent exercises. I think it's tomorrow. But if you wanted to enrich the 73 genes that you know are upregulated in response to growing in blood, you can always do this by clicking on the Analyze Results, and you'll be presented with a few options how you can enrich your analysis further. And so, for example, if you click on the gene ontology enrichment, you'll be enriching for any Go terms that happen that are enriched within the 73 genes returned by your search in step 2. I have pre-run this previously, and amongst these genes, there was an interesting observation that I have enrichment for the genes that are important for the host pathogen interaction, possibly. And while the window will quickly, hopefully, refresh, I just wanted to show you how this table will look like. Here we go. So I've analyzed these genes based on biological process, and just quickly looking at the results. Some of the genes, 42 out of the 73 in total, have a go term associated with biological process for interaction between species. So for me, this was quite an interesting observation that posed a question whether perhaps I should look at the host pathogen interactions, and specifically host data, when Candida interacts with either human cells or other models for human disease. And coincidentally, we do have a few data sets in FungiDB that integrates the host parts of the dual data sets. And in this case, if you search for the, say, data sets that contain blood, I can identify that there are additional data sets where we can look at the host transcriptome responses in terms of when a human whole blood is incubated with various Candida species. And so in this case, you could have an approach that uses not only interrogation of the fungal specific data, but also host responses to infections with fungal pathogens. And you would use the same strategy as I have shown you before, and as you would learn throughout this workshop, to apply to different data types and data sets of FungiDB or any other databases. Okay, so I think this is it for me. Let me know if there are any questions.